Hello, this is Dr. Ricks, and today I want to invite you into a different way of thinking about genetics. So this is epigenetics, the science of how our environment, experiences, and even love can shape how our genes function. So by the end of this talk, you should be able to answer three questions. One, what is epigenetics? Right? What is epigenetics? What role does chromatin play in regulating gene transcription? Three, how do specific proteins remodel chromatin to turn genes off and to turn genes on? So to get there, we're going to follow a story. And I think by the end of this, you'll never look at inheritance the same way. So this is my mom, and for most of you, you haven't seen me, let alone my mom. But today you're gonna meet my mom and me. This is my graduation day. I graduated from Moore School of Medicine, and this is the day that I graduated. I bring up my mom here because I believe that the way she raised me, the way she cared for me, the way she nurtured me throughout my life has a huge impact on the way that I deal with life now, the way I deal with stress, the way I respond to stressful situations, and the way I handle things is a direct reflection, at least to me, I believe, is a direct reflection of how my mom raised me. I want you to think for a second. For you, just reflect. How were you raised? If you're a parent, how do you raise your children? As we'll see throughout this presentation, that it may have some impact on the way they turn out and the way they behave, and it shapes their habits and behaviors, and it may be shaping their biology. In the animal world, especially in rodents and rats, mothering behaviors can be measured really clearly. Mother rats show affection through three actions: licking, grooming, and archback nursing. Some rat mothers are high lickers. They shower their pups with attention. Others, not so much. They provide minimal grooming and interaction. But does this difference matter? And what does this mean for the pups? Well, scientists ran a long-term study. There were two groups of rats. They studied them for the first 7 days of their life. Some rats were put with high licking rat mothers, and others were put with low licking rat mothers. And they followed them into adulthood and studied their behavior into adulthood. Here's what they found. In the highly licked pups, those pups were calm, easy to handle. And when they say easy to handle, remember, we're in a lab. And so you have rats that you go to grab. You have to, grab them and handle them in studies. And so you have to grab the rats and let them kind of walk around and move them from cage to cage. And in those situations, a calm rat or easier to handle rat means that you can kind of put your hand in the cage. You still have to be, careful, but you can put your hand in the cage, grab the rat, and then move the rat around with no major issues, right? However, sometimes that's not the case. And you have to be very careful when dealing with the rats. And so for a scientist, if you're, in a rat study, you are happy when you have rats that are calm, easier to handle, and respond to stress well. So you're like, oh man, I can't wait to go to the cage today. You maybe see rat 35, right? And this rat, it'll be calm, easy to handle, and respond to the stress. And the stress is me kind of putting my hand in the cage and interacting and engaging. They respond to that stress well. I mean, not this well, no rat. It does this in the labs, but it kind of feels more like, I can deal with this rat a lot, easier. However, there's another group of rats. And in this group of rats, they were lowly licked as pups. So in the first 7 days of their lives, they were lowly licked. Those rats were anxious, jumpy, and struggled under pressure. So same species, same environment. I'm going to show you a different picture of a different rat. So imagine you putting your hand into a cage and you're about to get your rat and you see this. That's like, oh, right. You want to, it's a little bit more anxious, right? And you see it's a little bit more jumpy. In this particular case, they weren't, two different types of rats. The pictures were showed up, but they weren't. I just wanted to show you an image here. But they were the same species, same environment, but different maternal care, and showed a drastic difference in behavior. That difference didn't stop at behavior. Researchers dug deeper and measured the gene expression, especially for the glucocorticoid receptor gene or the GR gene. So remember that gene, we'll be looking at it throughout the course of this, the GR gene, which plays a role in how we respond to stress. In highly licked rats, the GR gene expression was significantly higher than in the low lick group. This gene helps to shut off the stress response. So more expression means faster recovery from stress. But that raises a major question. How can maternal licking in the first 7 days of life change gene expression? We're talking about genetic activity, not just an emotional effect. How could nurturing be rewriting nature? So let's take a step back and talk about stress. When we perceive danger, the brain kicks in the stress response system, the HPA axis. Cortisol is released and travels to the brain, where it binds to GR receptors. Remember, we talked about the GR gene, GR receptors, the glucocorticoid receptors. So cortisol is released, travels to the brain, and binds to those GR receptors. Once enough cortisol is detected, the brain gets the signal, hey, we're okay, we're safe, and shuts off the stress response. So fewer receptors means weaker detection. So the stress circuit stays active longer. That's not ideal. So again, what determines the number of receptors? And why did those levels differ based on how much the pups were licked? I want you to take like 30 seconds or a minute or so and write down your best hypothesis. How could a behavior like licking and grooming result in a measurable, lasting change in gene expression? What could be the link between care and this transcript elevation? At this point, feel free to pause the video and then unpause the video when you're done writing down your thoughts. All right, let's get technical for a minute here. We measure the expression of the GR gene using the RT-qPCR. The RT-qPCR detects the amount of mRNA being made. So we're asking, how actively is the gene being transcribed? But transcription is just one piece. Many things can influence gene expression. Transcription may not happen at all. And if transcription doesn't happen at all, that's here, transcriptional regulation, where transcription doesn't happen at all. The mRNA might not be processed properly. Remember, we talked about the RNA processing, so 5' capping, splicing, the addition of the poly A tail. That may not be happening much either. That's RNA processing regulation. That can have something to do with how much mRNA is available because we're measuring mRNA. The other thing is the RNA could be degraded before it's made into protein, or the RNA could be actively being degraded. That's another form of regulation as well. These are all layers of regulation, and any one of them could explain the differences we're seeing. Remember this slide, though, and go back to it and study this slide. This slide is key when we talk about gene expression regulation and the points of possible regulation. So as you see, transcriptional regulation, RNA processing regulation. There could be regulation in mRNA localization and transport. You'll learn about that a little bit later, but understanding that that is a point of regulation is key. Another thing is translational regulation, going from mRNA to protein. There's possible, there's regulation that can happen there. Post-translational modifications matter when we talk about active protein and deactive protein, and then regulation of protein degradation matters as well. So when we talk about post-translational modifications, that's phosphorylation and things like phosphorylation. You'll learn about that later, but just remember it for now. And then proteins going into degraded proteins, that's ubiquination, and you'll learn about that as well. So just remember this slide, keep it, study it. You'll need it for the exam. Now let's get back into the case. All right. So let's return to the central dogma of molecular biology. DNA is transcribed into RNA, which is then translated into protein. We aren't looking at the protein levels here. So when we looked at the a gene expression or the mRNA expression, we're right in the middle step. We're not at the protein step. So if the gene wasn't being transcribed properly, we'd see less mRNA, just like we did in the low-lick pups. But could this be a mutation in the DNA? That's where we'll go next. Okay, so we're seeing huge differences in the GR gene expression, but is it caused by mutation? So let's look. Here's the actual DNA sequence for exon 1 of the GR gene in both groups. The top line is query, which is the low-lick rats. The bottom line is the subject, which is the high-lick rat. And we're just going to look for differences between the base pairs. Looking at the DNA, maybe this can help us to see if there's an issue with the DNA leading to an issue with the mRNA. As you can see here, 115 out of 115 base pair match, no gaps, no substitutions, no differences. So this is not a genetic mutation that's causing what we're seeing. The gene is intact. So we have to ask, if it's not the DNA sequence that's changing, then what is? Well, we know from central dogma DNA comes from RNA. So if the amount of RNA we're seeing is different, maybe something's stopping or interfering with transcription before the gene ever gets expressed. This leads us to explore beyond genetic and see if there's another cause of this result. So if we zoom in here for a second, look at DNA, let's talk about just the characteristics of DNA. So DNA in your body is long. Think about this. In a human, there's approximately 10 trillion cells. So if you were to line all the DNA found in every cell in the human body, it can stretch from the earth to the sun 100 times. If we go into one cell and stretch out all of the DNA in it, it could be as tall as a 6-foot male. We've talked about this in our DNA condensation videos, right? So how do we take all that DNA and fit inside a very, very small space? And it's through DNA condensation. But let's revisit it. So DNA, which is very, very long, wraps around histones. And that's how we start to condense it or get it to decrease in its size, right? So for a visual example for yourself, what I would like you to do is take out your power cord on your laptop just for a second, right? And I want you to take the power cord and try to fit it inside of your bag, but not the big portion of your bag. Like maybe there's a small pocket in your bag, right? In order for you to get that long cord into a small space in your bag, you have to do what? You have to wrap the long cord around the base of your charger. And that's what we always do, right? We take the long cord, wrap it up, and stick it inside of a small space in our backpacks. All right. Same thing with your DNA. We're going to take this long stretch of DNA, and we are going to wrap it around these histones. And as we wrap it around the histones and continuously do that, then we'll pack it up until it's in a form, a chromosome form, that where it can fit easily inside of our nucleus. So let's go through this again, right? Let's make sure we understand each part. So we have DNA. That DNA wraps around the histones. The histones are made up of a core of 8 molecules. So it's 2 of each. So it's H2A, H2B, H3, and H4. From your DNA condensation videos, somebody may say, well, where's H1? So go back to that video, make sure you know where H1 is too, because it matters, right? But here, H2A, H2B, H3, and H4, that's the 2 of each, make up the core of the histone molecules. DNA wraps around that, forming the nucleosome. That's the first part of this DNA condensation. From there, the DNA and the histones interact really tight. They form the 30 nanometer chromatin pad, then 300 nanometer, and then consistently condense until we get to 700 and then a full chromosome. Well, this interaction between DNA and histones is more than structural. It's regulatory. How tightly or loosely DNA is wrapped determines whether genes are accessible or inaccessible. And here's the key idea. If a gene is buried in tightly packed chromatin, it can't be transcribed. So gene expression isn't just about the gene, it's about access. That's what epigenetics is all about. When we look at epigenetics, we say, okay, what does epigenetics mean? It means above genetics. The epi part means above. So it means above genetics. And epigenetic changes can happen due to stress, due to diet, changes in environment, exposure to toxins, and even trauma. So let's explore these epigenetic changes. So we said epigenetic changes are really about DNA interacting with histones. That interaction matters. Let's explore how that interaction is modified, changed, and ultimately impacts gene expression. And I just want to really break down this whole concept of transcriptionally active chromatin versus inactive chromatin and what can lead to those things. I'm going to go through every part of this diagram before we start to add to it, okay? So what you see on the screen, I want to break that down and then we'll add to it a little bit. First off, the green spears that you see here, these green spears are the histone cores. Remember, it's two each of four histones, H2A, H2B, H3, and H4. And so here in this diagram, you see a spear here, a spear here, and a spear there. These are all the histone cores, okay? Now, the purple strand that you see represented here and here, this is the DNA. And so remember that this is all about how DNA interacts and engages with histones. And so we see DNA here, here, and down here, where the purple strand is always the DNA. The other part of this are the lightning bolts. So the lightning bolts that you see here are representative of histone tails. And there are tails on all of these histones. As you notice, one of the most important ones is a tail coming from histone 3. If you notice there's Ks, those are lysines, like K4 and K9. Epigenetic modifications like methylation will occur with the addition of those methyl groups being added there. So we have histone tails on the histones, and we have, so we have histones, we have DNA, and we have histone tails. All right, awesome. Now, this is key. DNA is negatively charged. Write that down. It matters. DNA is negatively charged. These histones are positively charged. And if you know, opposites attract. So this positively charged histones and these negatively charged DNA, they interact very tightly. They have a high affinity for one another. Naturally, they bind tightly. When they bind too tightly, it blocks access to genes, and transcription can't happen. So gene expression, remember gene expression is a measure of how much a gene is transcribed. If we can't get to the genes, we can't transcribe it. So gene expression goes down. That's what you're seeing down here, where you see a tight interaction between DNA and histones, and these genes are not easily accessible. Look at the space between this nucleosome and this nucleosome, or this one and that one. And you see that it's hard to get to these genes. Remember, in order to see gene expression, we have to get to the gene. What needs to get to the gene? RNA polymerase and all the transcription factors. There's a big complex that needs to get to these genes. There's not enough space here to get to this gene. There's not enough space here to get to this gene. However, there is space up here. Look. See this nucleosome, that nucleosome? Look at all that space. Boom, get in there, transcribe that gene. Boom, get in there, transcribe that gene. Okay? So all RNA polymerase and all the transcription factors can get in here and transcribe the gene. So down here, you would have a decrease in transcription. Up here, increase in transcription because the genes are more accessible. This is looser chromatin, tight chromatin. transcriptionally active chromatin, or euchromatin, and transcriptionally inactive chromatin, or heterochromatin. All right. So what causes this loose interaction and what causes this tight interaction? Well, there are ways to modify the histones where we can lessen the charge or lessen the positive charge so it interacts more loosely with the negatively charged DNA. Or there are ways to interact with the DNA to make it interact with the histones a little bit less. So how can we change from transcriptionally active chromatin to inactive chromatin, or vice versa, from inactive chromatin to active chromatin? And that's where the modifiers come in. What are the modifiers? So get you're writing down these notes. It's another part to write some notes. Let's talk about these modifiers. The first modifier we'll talk about, and modifier means it's the enzymes that will modify this interaction. The first modifier we'll talk about is histone acetyltransferases, or HATs. Now, what do HATs do? And I want you to notice something. If you notice at the bottom in this key here, the acetyl groups are what we're adding, and notice where we're adding them to the histone tails, right? So these are acetyl groups being added to histone tails, and they're being added there by a group of enzymes called HATs. Now, HATs matter, right? So, what are the HATs? They're histone acetyltransferases. And what they do is in the name. So, if you ever want to know what these enzymes do, reverse the name, right? It's a histone acetyltransferase. So, what do? They transfer acetyl groups to histones. And that's exactly what that is. They add acetyl groups to lysine tails, decreasing the histone's positive charge. This loosens the DNA histone interaction and makes the transcription easier. So these HATs, or histone acetyltransferases, can add acetyl groups to these tails. And once that happens, loosens that interaction. So you go from transcription inactive chromatin to boom, active chromatin. So HATs make things loose. And that makes sense. Think about it. Whenever you're going out somewhere, or somebody's around, if somebody has on a hat, they're a little bit more loose, right? So I put on my hat and I'm a little bit more chill. I put on my hat to chill out, right? So HATs help to loosen things up. Okay? Now, there are other proteins that do the absolute reverse of that. They're called histone deacetylases. And what do? They deacetylate histones, meaning they take acetyl groups away from histones. When they do that, it results in transcriptionally inactive chromatin. Because what they're taking the acetyl groups away, bringing back that positive charge, making the interaction between DNA and histones more tight. That suppresses transcription. All right. Now, there's another thing we can do. So we just talked about how we could impact the histones. Well, you can also impact the DNA. You can impact the DNA through enzymes called DNA methyltransferases. What do? Remember, reverse the name. So DNA methyltransferases transfer methyl groups to DNA. They add methyl groups directly to DNA. When this happens, DNA becomes harder for transcription machinery to access, and the genes get turned off. So DNA methyltransferases can cause transcriptionally active chromatin to be inactive chromatin just by adding methyl groups to the DNA. So we looked at a way to impact the histones through histone acetylation, and now we've looked at a way to impact the DNA by adding methyl groups to the DNA. Now, there are histone acetyltransferases. We know what they do based on the name. They transfer acetyl groups to histones. Okay. Then there are histone deacetylases. They remove those acetyl groups from the histones. Then there's DNA methyltransferases. They add methyl groups to DNA, causing transcriptionally inactive chromatin. Is there a possible way to remove the methyl groups from the DNA?
Yes!
They're called DNA demethylation enzymes, and those demethylation enzymes remove methyl groups. And what do you think the result would be? The result is transcriptionally active chromatin. All right, so hopefully you get that. Let's review. Okay, now DNA is negatively charged, histones are positively charged, so naturally they interact very tightly. There are enzymes that can help us to lessen that interaction. HATs are one of those, where HATs, what they do are histone acetyltransferases. They add acetyl groups to the histone tails and cause that interaction between DNA and histones to loosen up, resulting in transcriptionally active chromatin. Okay? There are other enzymes called histone deacetylases that remove those acetyl groups and tighten that interaction between DNA and histones and cause transcriptionally inactive chromatin. Those enzymes are histone deacetylases. That's the way we can interact with the histones. Now, there are ways to modify the DNA too. Modification of the DNA happens through DNA methyltransferases. What do? They transfer methyl groups to DNA. Upon doing that, they tighten the interaction between histones and DNA and cause transcriptionally inactive chromatin. So, loose interaction between DNA and histones, transcriptionally active chromatin, meaning more gene expression because more transcription. Tighter interaction between DNA and histones, transcriptionally inactive chromatin, less gene expression because there's less transcription. Okay, go over that a few times if you need to. Pay attention to each one of these parts. They matter. I'm going to now get into breaking down each one of these a little bit more and give you a little bit more detail before we try some exercises. All right, let's get it. Okay, so let's look at epigenetic control, DNA methylation. The DNA molecule itself can be modified by methylation. So DNA methylation occurs within these places called CPG islands. Write that down, you'll need it. They're regions of the DNA that have high frequency of cytosine and guanine, dinucleotide DNA pairs. So remember in complementary base pairing, you see C and G, right? So A and T are paired, and then C and G are paired. Wherever you see a high frequency of C and G, those are called CPG islands. And DNA methylation typically occurs within those CPG islands. So the cytosine base of the C and G pair can be methylated, which causes the genes to be silenced as transcription factors binding to the promoter is blocked. Remember, it tightens that interaction. Now let's add one more type of modification. You didn't see this in the big graph, but I'm going to add one more to your knowledge bank here, and it's histone methyltransferases. You're like, okay, we've talked about histone acetyltransferases, we talked about the hats, and we talked about the deacetylases, but we haven't talked about methylation on the histones. What's this about? All right, so methylation can occur on those histones as well. Where do the modifications normally happen when they happen to the histones? They happen on the histone tails. So these enzymes add methyl groups to histone tails. Now, this is tricky though, because with hats, whenever the hats add acetyl groups to histones, then immediately we know activation, right? We're going to see an increase in gene expression. We know that. Hats make things loose. We know that, right? Whenever you remove those acetyl groups, then it's boom. We know for a fact that is repression, right? Or it's going to result in a tighter interaction between the histones and the DNA or the heterochromatin. Boom, we can see that. We know that based on the graph. But what happens with histone methyltransferases are a little bit different because it's determined by where the methyl group is added. For these histone methyltransferases, if the methyl group is added to the fourth lysine, well, that causes activation or a lesser interaction between the DNA and the histones. Looser interaction. However, if the methyl group is added to lysine number 9, then that causes repression. And so with histone methyltransferases, you have to remember that it can either be repressive or activating based on where the methylation occurs. So jot that down. That adds another layer of complexity here when we talk about epigenetic control. So H3K4, remember we said lysines are noted by Ks. So it's on histone 3, lysine number 4. If you have methylation there, activation. Histone 3, lysine number 9, then it's repression based on where the histone methyltransferases add the methyl groups. Okay, cool. For DNA methyltransferases, it's less complex. It's add the methyl group to the DNA. These DNA methyltransferases, what do? It's in the name. They transfer methyl groups to DNA. Once that happens, we know the result of that. The result is always transcriptional silencing. All right, y'all. Let's see if we get it. Let's see if you get it. Let's see if you understand. It's game time. What time is it? Game time. All right. We had our study time. Now it's game time. All I'm going to do is I'm going to play something for you. I want you to look at it and then tell me, what do you expect? Do you expect gene expression to increase because of what happened on the screen? Or do you expect gene expression to decrease or repression? So it's either activation, which means more of the gene is being transcribed, so activation, or repression, less of the gene is being transcribed based on what we'll see on the screen. All right? So hopefully you have the rules. Our thing is always activation or repression. Loose chromatin, a loose chromatin, a euchromatin, heterochromatin, or tight chromatin. All right, ready? Here we go. Now, I want you to look at the screen and really determine what's on the screen. I'll give you a clue here for the first one. You have HAT, which are the histone acetyltransferase. It's carrying that acetyl group. This is always the histone tail. Remember the histone tails? You have lysines on that histone tail. So we're looking at H3, and now I'm going to just play it, and you tell me, what do you expect? Either gene activation or repression, based on what's going to happen here. Here we go. Okay? Activation or repression, based on what we're seeing, based on what you know, based on your notes. What do you expect? Okay? I saw a loose interaction. Boom! There you go. Gene expression, genes on, activation. Because when hats do their thing, they make things loose, and loose will give us this, right? Loose means that we'll have gene activation because here is your gene, and all your transcription machinery can get to this gene because of the loose interaction. Hats make things loose. When things get loose, things get active, and we can transcribe that gene. Awesome. All right. Now, here's another one. Okay. Just look at the screen. No explanation here. I'm going to play it. And once we play it, I want you to say activation or repression based on what happens. Here we go. Activation or repression. What you got? Boom. All right. You should have said repression here because that is a DNA methyltransferase. What do? It's in the name. Transfer methyl groups to DNA. And when that happens, what happens here is you have repression, heterochromatin. That's what happens when DNA methyltransferases add methyl groups to DNA. All right. Let's try another one. This is a tricky one. Remember, we have HMTs on the board here. So the histone methyltransferase, what do? It's in the name. They transfer methyl groups to histones. All right. Now, but based on where that happens, you have a different effect. So I'm going to play it. You tell me, what do you expect based on what we studied? Activation or repression? Boom. Activation, right? Because when methyl groups are added to H3K4, that's what we're seeing here, H3K4, that's activation. What if it would have landed on K9? That's repression. All right. So when we deal with the histone methyltransferase, it matters where the methyl group is added. Okay, awesome. Let's try one more here. What do you expect? Based on what just happened, what do you expect? Activation or repression? Let's go. I know you got it right. I'm just assuming you got it right, right? It's repression there. What happened? There you saw HDAC, which is a histone deacetylase, remove the acetyl group from the histone tail, leading to repression. Now that we understand genetic mechanisms, DNA methylation, histone modifications, chromatin accessibility, let's go back to our original mystery. How could maternal licking behavior in the first 7 days of a pup's life influence the expression of the GR gene and ultimately that pup's behavior? Take a moment, write down your hypothesis. What could be going on here?
Here's the webpage, and the first part just reads, some mother rats spend a lot of time licking, grooming, and nursing their pups. Others seem to ignore their pups. Remember, pups is just the word for baby rat. Highly nurtured rat pups tend to grow up to be calm adults, while rat pups who receive little nurturing tend to grow up to be anxious. It turns out that the difference between calm and anxious is not genetic, it's epigenetic. The nurturing behavior of a mother rat during the first week of life shapes her pup's epigenomes. And the epigenetic pattern that mom establishes tend to stay put even after the pup becomes an adult. So down here, we're going to click lick a rat pup.
Deep in the brain of a newborn rat pup, methyl molecules, green, silence the GR gene. When it's active, the GR gene produces a protein that helps the body relax after a stressful event. The type of care a pup receives from its mother during the first week of life can change the expression of this gene.
So I'm going to start to run the simulation, but as I click start and the mom rat will appear and she'll start licking the baby rat, the pup, I want you to keep your eyes peeled on what's going on in particular over here. All right, so the first part was to lick the rat a lot, as much as you can. So here we go.
Wow, you're an attentive rat mother. It's surprising your pup has any fur left on its body. Your licking and grooming has seriously activated your pup's GR gene. Your pup will have an easy time relaxing after stress. Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
So I'm hoping that you noticed that as we licked the rat pup, the GR gene slowly started to uncoil, and at first it was like very tightly wound together, but now it's much looser, easier to access those genes. So we're going to click lick another rat pup, and then this time we're only going to lick it 3 times. I want you to continue to look over at this box to see what happens to the GR genes. 1, 2, 3.
It looks like you had better things to do, since you didn't lick and groom your pup very much. Its GR gene is still inactive. Your pup will have a hard time relaxing after stress. Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
So in this video, you have your GR gene here. As the mother is licking, you see the DNA and the histones having a looser interaction. Well, what's going on there is DNA is being demethylated. And so you see methyl groups leaving the DNA, allowing for there to be a looser interaction between the DNA and the histones. That's what's happening here when the rat mother is licking in those first 7 days. She is changing the interaction between the DNA and the histones here in those 7 days. When she licks only 3 times, then you see less, you see the green dots here? You see less of those methyl groups leaving the pup. And the less methyl groups that leave, the more that GR gene is still going to be transcriptionally inactive because the DNA and the histones are still very tightly interacting. So when you have the loose interaction because of the excessive licking, then you have a lot of GR gene being transcribed because it's a loose interaction between histones and DNA. If you have low licking, then you're going to see the complete opposite of that. So this is a different lens, a different way of thinking about how traits are passed on. Traditionally, we think of inheritance as strictly genetic, DNA passed through sperm and egg, right? But here we can see that a mother's behavior rewrites the expression of a gene, shaping behavior, impacting physiology, all without changing the DNA sequence. This is the power of the epigenome and the power of epigenetics. So the question is, does this happen in humans too? We see it in rats, okay, but does this happen in humans? And the answer is yes. So we've seen epigenetic impact of the GR gene in human brains with children who have suffered child abuse. So the question is this real? Like, is this a real thing? And what I see is what I'm seeing real? Yeah, it's real. This is the paper. I put the reference here. I put the paper here. You can go and find the paper and read the article for yourself. And so you can get a good gauge of what is going on here. This is a true paper and a real story. The epigenetic marks that are seen in children with child abuse, that's a real story as well. And so not only are we seeing this in rats, but we're seeing a epigenetic impact with children who are suffering child abuse. And this is that paper. You can go and find that as well for more reading. Another interesting thing is what's happening in terms of inheritance. And what do I mean by that? And so they studied the grandchildren of Holocaust victims and looked to see if there were any epigenetic impacts. And what they're seeing is that the grandchildren of Holocaust victims are showing signs of PTSD, which means that the things that your grandfather went through, the things that your mother went through, could possibly be impacting the way you behave in certain situations. That's a way different way of looking at inheritance, not just what happens, not just, the genes that are being passed down, but the epigenetic changes that happen because of trauma, because of stress, because of environment. Those epigenetic changes not only impacts that generation, but could it be impacting you? This leads us to think about questions like, well, my ancestors went through slavery. What happened epigenetically to the ancestors that went through those harsh conditions? And how did that impact the generation after them and me? It's a different way of looking at environment and how it impacts genes.
So our genetic code, this is our DNA, this stays the same. And the cool part, it's the same in every cell. Our genetic code is the same in every cell. But we have eye cells, we have liver cells. We don't have eye cells growing in our liver, hopefully, and vice versa. And this is because we have epigenetic markers, same as the epicenter and the epidermis. Epi means above. So epigenetics means above the genome. These are big proteins, big markers that sit on the genome, and they say, nope, don't read this part of the genome, or come read this part of the genome. And they change the way that DNA is wrapped around histones or sit in a space.
So it turns them on and off. Is that a way to, can we think about it that way?
You can, yes, I think you can.
So the expression of the gene
Expression,
itself
that's a better word, expression.
is dependent upon the epigenetic determination?
How much is expressed and when it is expressed depends on the epigenetic environment.
So here we have transgenerational epigenetic inheritance. How does that get inherited?
Oh my goodness. I mean, if we all knew how it got inherited like that quickly, I think you'd be addressing me in my Nobel Prize. But since we're not there yet, but there are questions surrounding that.
Promise here, when you get your Nobel Prize, you're coming back here on the show.
Yeah,
okay,
exactly.
All right, Girl Scout honor.
Okay.
So there's many questions that are left unanswered when it comes to how transgenerational epigenetic inheritance works. I think we're at the space now in neuroscience where we are observing that intergenerational inheritance of an experience is pretty much canon now. We can say with my studies as well as others that an experience in a parent will change the way the offspring develop. And I'll be pretty specific with this.
Experience in a parent.
Experience in a parent.
Even before the child is conceived.
Exactly. Before the child is conceived.
Let's talk about this, and you can go and check out Ghost in the Genes. That's, something you can really look at if you're interested in this topic. But let's see if we hit the things that we are trying to get to know today. All right. So we said by the end of this, you would know what epigenetics is. Hopefully you get that. Epigenetics is a change in phenotype without a change in the genotype. What I mean, there's no change to the genes. We see there's no change to the DNA, right? However, the behaviors are changing, gene expression is changing. A lot of things are being impacted. However, the genes itself, the A's, the T's, the G's, and C's are not being impacted at all. Epigenetics is above that, right? Epi meaning above is above the genetics. So we see a change in the phenotype of the behavior with no change to the genotype. What role does chromatin play in regulating gene transcription? We see loose and tight interactions between DNA and histones can alter gene expression. Loose interactions, high gene expression; tight interactions, low gene expression. We see loose interactions, high gene expression; tight interactions, low gene expression. How certain proteins can control chromatin remodeling. Now we know that they are histone methyltransferases. What do? It's in the name. They transfer methyl groups to histones. We know that they have an impact on gene expression. Histone demethylases. What do? We know it's in the name. They take away methyl groups from histones. How does that have an impact on gene expression? HATs, or histone acetyltransferases, right? What do? Yeah, I know you said it. You said it's in the name, right? And they transfer acetyl groups to histones. We know that has an impact on chromatin remodeling. And the deacetylases, we know they have an impact along with these DNA methyltransferases. Now we know all of these different types of chromatin remodeling enzymes. And these chromatin remodeling enzymes play a major role in our understanding of epigenetics. Hopefully you got something from this. Hopefully you feel confident in understanding this a little bit more. If you do, that's awesome, right? That's exactly what this is all about. Take your time with this, pause it, rewind it, go through it again, whatever it takes. But I want you to get this because it's key for your exam coming up. But not only that, but just for your scientific knowledge. You took an epigenetics course. That should mean something. It should mean you have some understanding of epigenetics, right? That's what that means. You took the course because you wanted to leave with some understanding of epigenetics. This is a major part of the base of that knowledge. From here, you can get into the way more complex things in epigenetics, right? However, this base knowledge will help you to get to where you want to go, at least to say, hey, you know what? I understand this stuff. And when you hear people talking about the crazy work that they've done in the field of epigenetics, you'll have a base knowledge to follow them and understand their great discoveries, right? And there's going to be a lot of epigenetic discoveries. I've talked to plenty of scientists who are really involved and engaged in this. So hopefully you feel like you have that. If you are at this point in the video and you're like, what? What is this about? I'm very lost. Go back, look at it again. Take the journey for real. Take the journey again. And if you're still lost after that, talk to me. I will walk you through this slide by slide, and we'll get to the point where you have a full-fledged understanding of epigenetics and epigenetic control. All right. Y'all have a great day. Bye.